In this episode, we speak with Dr. Ani Deshpande from Sanford Burnham Prebys, whose research focuses on the epigenetic and transcriptional regulation of cancer. By dissecting how oncogenic signaling pathways rewire the chromatin landscape and transcription factor networks, Dr. Deshpande aims to uncover vulnerabilities in tumor progression and resistance. How do epigenomic alterations sustain malignant cell states? Can targeting transcriptional dependencies offer new therapeutic avenues? Stay tuned as we explore the interface of cancer biology and precision epigenetics.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Today I'm joined by Dr. Ani Deshpande from Sanford Burnham Prebys. Hello Ani, how are you doing today?
I'm fine, how are you?
I'm doing good. I'm very happy that this came into place. What have you been up to in the last couple of days in terms of maybe lab work or traveling? Is there something that you can share?
Oh, we have been doing some really cool stuff in the lab, and I've been, the graduate students mostly have been sharing interesting data, and it's always one of the pleasures of going into the lab and, the students come and show you a piece of really cool data. So that's what I've been up to the last 2 days.
So to focus more on you, a question I like to ask every guest to start off the podcast and ease into the topic is, how did you become interested in biology in the first place? And then maybe a little bit later in pursuing a career in science?
Well, interestingly, I was actually, no, I don't remember being really interested in science per se growing up in Mumbai, in India. I was more interested in literature, in languages. And in fact, one of these tests they do, the aptitude tests or what you should do in your career kind of tests, I ticked some boxes and gave that sort of exam, and they suggested that I should be a linguist. But in those times, 1990s India, linguist was not a profession that people thought would help you earn a living or something like that. And so, of course, I was very curious. I was always very curious about things. I used to keep asking questions, why and how and stuff like that. But the real moment in my life, and of course, my love for literature took me to reading. I read a lot of science fiction, which I really got fascinated by. And then the real turning point for me was when my sister enrolled in a bachelor's degree in microbiology. And she used to come and tell me these fascinating stories of the discovery of DNA and antibiotics and all of these. And as a teenager, you're not really interested in listening to your siblings or even, following their advice. But I was completely captivated by these stories. And I was like, wow, this seems amazing. And then I just sort of followed her in enrolling for bachelor's in microbiology and then a master's. And I was totally hooked. And one thing I realized is that as I read more and more of scientific discoveries and the process and the stories, what I realized is, as I think either Richard Dawkins or Neil deGrasse Tyson, one of these say that, and I'm paraphrasing, they say that it's not just exciting, but it has the added advantage over science fiction of also being true. And so I started loving those stories. And then I, of course, decided to pursue my PhD. And that's how I ended up actually in Munich, not too far from where you are, Stefan.
Yeah, that would have been my next question, right? You started your research career in Munich. And what made you go there?
So at that time, I was applying to a lot of places in the United States. And then I got a chance to go to Germany. And that was a time when there were not too many Indians actually applying for PhD positions in Germany. But Germany had just, I think, made a decision that they are going to invite a lot of international students for their PhD programs. This was in the early 2000s. And we were really some of the first. And I joke always that when I went there were so few Indians in Munich that we had a very small community. We knew each other. I say, it's like the Mayflower. And so it was very tough, of course, coming from Mumbai to Munich. It was two completely different worlds. I had a lot of challenges just, uh, dealing with the language and the culture, very different. And then the good thing was that the labs, everything was in English, and some of us did learn German, although you didn't need to for the lab environment. But it's always good to learn German to interact with the colleagues. And then our lab had a lot of Italians and Germans and Greek and Indians and a lot of people from all over the world. So it was my first exposure to an international lab environment, and it was absolutely fabulous. I learned so many things from that. And then fell in love with Munich because it's such an amazing city to live in. And, I did not have a car, so we always went everywhere with public transport. And I just loved that. And the science there was exciting. I started the, after a brief period at the Max Planck Institute, then I started at the LMU, the Ludwig Maximilian University, as you know. And so at that time in university clinic at Grossharden, which I'm sure you know, Stefan, there was a monumental effort going on in research on AML. They were doing extremely good stuff, one of the best. They had a lot of clinical studies that were very important, seminal studies on the different mutations that were being found. And the uh, working there in that environment was really good because you were not only exposed to the research there, a great research environment. Actually, I was at the Helmholtz Center building near the University Clinic in Munich, and the but also the exposure to all the patients. We always went there for lunch, and we saw all these clinicians really busy working Uh, with the patients and then coming back to the lab. So that was my first time actually working closely. Many of my colleagues, my mentors, my were physicians, and so it was wonderful to see that how the work that you're doing directly is related to the patients that you. I didn't get to see the patient, but it was a great environment to work in.
So you already mentioned the AML, and you developed several murine models for refractory AML subtypes. Could you walk us through the significance of these models and how they've advanced the therapeutic discovery?
So at that time, I was the first PhD student of Dr. Christian Buske at the LMU in Munich. And he had just come back from the lab of Keith Humphreys. And what they were doing at that time is that they were developing mouse models of leukemia. So you would discover either a mutation or a gene fusion, and then you would put it in mouse stem cells and make a bone marrow transplantation model. And that was sort of the in thing at that time. And they were doing that. And my mentor was Stefan Bolander, who had come back from the lab of Janet Rowley, as you know, the legendary Janet Rowley, and he had discovered a gene fusion with Martin Dreiling, and that was the CAM AF10 fusion. And it's a rare fusion, and it's seen in few patients, but it's associated with really uh, hard to cure leukemias and often seen in pediatric patients. And so they wanted to create this mouse model of the CAM-AF10 fusion. And I sort of got that task as the first PhD student, just come from India, not knowing much. But I was hooked on gene fusions in general. And I'm one of these people who start when given a problem, start thinking about, okay, what are gene fusions? What are the different types of genes that fuse, and why do they fuse, and what are the breakpoints? How are these breakpoints determined? So I sort of went down that rabbit hole, and in that process, I did develop the CAM-AF10 mouse model, and it had some characteristics that were very similar to the human leukemias. That was my first paper from my PhD. Of course, as similar as mouse models can be to human leukemias, and that helped me start thinking about gene fusions in general, then later worked on more fusions. Now, the one of the fusion partners of the gamma F10 fusion is AF10, which we didn't know what it does at that time, but there was research coming from the lab of Yi Zhang and others who had shown that AF10 is a chromatin modifier, a reader protein that associates with the histone methyltransferase DOT1L. And so I got interested in epigenetics. That was sort of my entry point for this looks interesting, because more and more reports were showing that epigenetic regulators were recurrently mutated in this disease that I was now interested in. And so that's what got me into writing an email to Scott Armstrong, who was then at Dana-Farber and Children's Hospital, Boston, to look for a position in.
Yeah, but I mean, this was not like one mouse model, right? Only you were involved in making several mouse models, right? So this was kind of fruitful PhD time, right? I mean, I got like one paper out of my PhD, but it seems like you were, very, very successful, and a lot of productive work came out of it.
So the paper that I published in my PhD, that was the CAM F10 mouse model. Was original. All the others that I have done later, they were not. It was not the first time that anybody had done that. People had done that before, and so I just developed them, replicated other people's work to make, to see if they are sensitive to epigenetic inhibitors and stuff like that.
Mm-hmm.
But the original development was of the CAM F10 mouse model.
Okay. And then you already said that you went to the US, right? So not the West Coast where you're now, but like the East Coast to Children's Hospital Boston, Harvard Medical School, and also the Memorial Sloan Kettering Cancer Center. So how did this come? And was this like two different postdocs, or was this like one, but you...
That is an interesting story, actually. So once I was interested in AF10 and DOT1L, I wanted to move to the United States, because eventually my goal was to actually establish my own lab, and the United States seemed to be a great place to do it eventually. And so I wrote to only one person actually for my postdoc, and that was Scott Armstrong. I had not applied to any other lab, and of course I prayed and hoped that Scott would accept me into his lab. And he was doing the lab. Scott's lab was actually not. So it was, of course, he was well established because he had published this very important papers, but it was still a relatively young lab, and so uh, he he offered me a position. I joined his lab, and it was one of the most important and exciting times of my life, because uh, all of my colleagues, they were all studying different epigenetic regulators, and Scott was an excellent mentor. And this was the time, as you know, that people were starting to think of epigenetic regulators such as methyltransferases like DOT1L, acetyltransferases, and others as potential therapeutic targets. And so there was a lot of excitement around that. And so I was very happy to move to Boston to Scott's lab and then start digging into it. And I made a conditional knockout mouse, something I wanted to do back in India when I had done an internship, and it was kind of interrupted. And then I got the opportunity to do that with the help of Stu Arkin's lab there. And so that was exciting. It was a big learning moment, and all my colleagues were extremely friendly people and very knowledgeable. And that environment really has informed my career to this day, and I remain in contact with almost all of them.
Oh, interesting. So what did you find out about DOT1L, the methyltransferase in leukemia, and maybe also about DOT1L inhibitors?
So what happened at that time is that when I reached Scott Armstrong's lab, there was a colleague, Catherine Byrne, who I think Scott placed me strategically next to her in the next bay. I shared a bay with her because she was working on DOT1L and I was working on AF10, the cofactor of DOT1L. And so it was a nice sort of partnership, try and understand how DOT1L and histone 3 lysine 79 methylation works in MLL leukemia. And there were a lot of other trainees in Scott's lab who were asking that question in different models and in different settings. And so Catherine found is that DOT1L is really important for MLL rearranged leukemia, and this has also been shown by two other groups simultaneously. And so that led to the idea that DOT1L could be a really good therapeutic target. And then as you know, in the this was the late, like 2010 onwards, there were companies who were starting to develop small molecule inhibitors of these epigenetic regulators, and not many had been tested in the clinic. And so there was excitement that DOT1L inhibitors are going to really make a huge difference. And of course, that didn't really pan out due to some limitations of the chemistry of the drug itself. But we do still believe that DOT1L is a very interesting target. In many experiments that we've done, even after that, we think that DOT1L is a very important and a very interesting target. But then we started thinking about other epigenetic regulators, and of course, Scott has worked on many inhibitors since, and you know the story of the FDA approvals and all of that. And when I started my own lab, I also started revisiting the question of what are the epigenetic regulators that are important for AML, and not just important for AMLs, but also perhaps selectively important for AML, and that will, as people in the field try to do, selectively important for AML cells and does not harm normal cells. So that sort of became the question that I started, one part of my lab started to address when I started my own lab.
Yeah, before going to your own lab, I have one other question because did you also work on AF10?
Yes, yeah. So most of my work actually in Scott's lab was on AF10.
Right.
So what he ended up showing is that AF10 is a protein that binds to DOT1L, enhances its enzymatic activity, something that we did in collaboration with Bob Rader's lab. Okay, so I did not answer your question about MSKCC, about Memorial Sloan. So what happened, I think 2.5 years after my absolutely wonderful time in Boston, is that Scott one day announced that he's moving to the Memorial Sloan because he had a position there. And so I actually did not want to move to New York because
that's is it in the middle of the city, right? In the middle of Manhattan?
Yeah. In the middle of my postdoc, I had to move in the middle of the city to New York. And I did not want to do that. So I just, trusted Scott that things will not, things will continue, and there won't be major disruptions. And thankfully, there were not many disruptions. So we moved to New York and continued the work there. Also, it helped me a lot that my wife, who was then a postdoc at Dana-Farber, she came and Scott offered her a position in his lab. So she also moved with me, and we both worked together in Scott's lab. And so we continued to work on AF10, and we showed that, and this was together with Bob Rader, showed that AF10 binding to DOT1L enhances its catalytic activity. And it is important for conversion of mono to di and trimethylation. And so the reason that is important is because we had shown that the conversion of mono to di and trimethylation is what is like a fine-tuning knob that regulates the expression of the HOX and MEIS and many other MLL target genes and other genes that are required for leukemias to continuously self-renew. And so we showed that if you disable AF10, you get an attenuation in the leukemiogenesis. So both in sort of normal cells and in leukemia cells, you see that this di and trimethyl mark goes away, but the leukemia cells seem to be kind of dependent on this. That was the work that we And so I published that paper from Memorial Sloan. Scott, as you know, later after working at Memorial Sloan, he's now back in Boston. But before Scott moved back, I got an offer to start my own lab, and I came here on a K-99 fellowship to the Sanford Burnham Prebys.
Yeah, maybe you can just continue on telling the story, because I guess from India to Munich, then to New York, and then to California now, I mean, your trip has always led you to be more west, right?
And yeah, I keep going west. I hope that I'm as far west as I can get. I'm not moving any more west here. Yes, you're right. Moving from the west of India and Mumbai to Boston, and then to New York, and then all the way to California. So after Memorial Sloan, I was looking for positions, and Sanford Burnham Prebys at that time was looking for someone working on cancer stem cells and epigenetics. So what I was doing really fit the bill, and so Robert Wexler, who was the program head, he interviewed me here. I came here, and for the first time, I saw. I came here to interview in December, and it was still very sunny, and I was wearing a half shirt. And coming from New York, I just felt, wow, this is amazing. This weather is like holiday weather, and this is just incredible. And I joked that I wasn't listening to any of the science he was telling me about, and I just decided to move here, which is, of course, not completely right. But, San Diego is a fabulous, fabulous place to do science and also to live in general. So that did inspire.
It's also nice to take a peek outside the window every once in a while and see the ocean, right? I mean, it's
yes. So we are like a mile away from the ocean, and so our lab is, and so it's really a very good place to live in, raise kids, and other stuff.
So when let me think for a bit, because one thing I was thinking about is that initially you were interested in literature, right? And like PhD, postdoc, and then when starting your own lab is really a period where you need to read a lot, right? I mean, it's not like trivia and science fiction, but it's still reading. So did this help?
Well, it actually, you'll be surprised, but it does not. In fact, I realized that the way we talk, the way we write is actually so far disconnected from the way normal, quote unquote, normal people talk. And this is, as I hope we will discuss at the end of, later in the interview, is that we actually, I think, do a really bad job in trying to communicate our science. Also, because scientific writing requires a very different, very rigorous, very exact, and use a lot of hedge words. You are very careful what you're saying. You use very dense technical language because you want to be sure of what you're saying, which is a great thing for science. But and so I spent the last 20 years reading those papers and going farther and farther away from the creative liberty that you can write with. I used to write poetry when I was in my teens, and I have never done that ever since. But the ways you do these two things are so vastly different from each other that you almost go in the opposite direction. And why that is important, I hope you will agree, is that when you then try to communicate your science, you are talking in a language that most general people do not understand. And that, I think, is a huge problem in today's times.
Yeah, because it's also that the things people work on are not like directly connected to clinics, because it's still pre-clinics and, so far away from the end use of what eventually comes out of it, that it's also probably very difficult to communicate in normal words.
Absolutely. And I think, we can't be experts at everything, right? When I did my podcast, as we'll talk about probably later, the reason I did that is because I realized that you can tell the story in a totally different way to connect these dots for people, is to tell them how whatever scientists are doing that may even seem like not important actually eventually becomes very important to the medicines they to the drugs they take uh, or to this
Mm.
their general lives.
Yeah, what I find nowadays is that you have those different parts in papers that need to be there because of good reasons. But if you look for informations, then it's very hard sometimes to find the information because you first look at one place, then it's not there, then you need to look at the other place, Uh, for example, the material and method section, right? How is it done? And then sometimes it's in the text, sometimes it's in this section, and then it gets very hard to find actually the information that you're looking for.
I think this is some of it is legacy, right? It's just the way things are formatted. I get frustrated when I get papers for review that are in the format that you're describing, where the figures are somewhere else, the legends are somewhere else, and it's totally frustrating. And I think that as a community, we need to really rethink, right? Yeah, is this the best way to do it? Should we continue to do that? And I think these are discussions that we are kind of avoiding, but they are necessary because it's making it more and more complicated for people to actually review any science. And there's a lot of fatigue then.
Yeah, definitely. So coming to the time when you started your own lab, did you feel or think that you needed to do something differently that you have done previously now that you can run the ship on your own? And what was it that you wanted to do?
Do you mean like in terms of science or the way you do science?
Both, but mainly the first question.
Okay, so in terms of science, I think that what I would have differently is that there are two ways of doing it. One is to do the things that you think will be fundable because the grant agencies are going to fund that science. And then there are things that you just find exciting. And, in the stories I tell, I keep telling stories of things that people have done, just, gone off in directions that they just thought were interesting and found something really um, remarkable and then led to some of the greatest discoveries there are. Of course, it has to be careful and carefully considered. But the problem in the second approach, where you do things that interest you, is that there are hardly any mechanisms that actually fund them. And I've struggled quite a lot with that. Half of my lab now does experiments that are important, but they are kind of like a template, right? That here's an epigenetic modifier, you think it's important, so do this experiment in vitro, in vivo, in you know the mechanism question, the omics and all that, right? The template, the set template where you basically fill in the details. It's important because they may go to clinical trials and then you want to know the biomarker, the mechanism, etc., right? But I get much more excited by experiments where you actually uh, do not follow the template and you do something completely different. The challenge has been that even the so-called innovation mechanisms, they're very conservative. I've been on those study sections and people just do not like those unless you've done them or at least done a substantial portion of them, which I kind of understand because they could be really key. But I think there need to be more mechanisms to fund ideas that are really very innovative, not following a template, and there just aren't any. So for a lab that just starts out like I was when I started out here, it's almost impossible to do that because you have to do things are going to get funded. So the big mistake I made is in the first 3-4 years of my lab, I just followed templated ideas that turned out to be boring. And so I would just not do that starting out. But again, some of that is not a choice.
Yeah. So is this why you now or in recent years focus on the development of tools and technologies, also using like CRISPR-Cas9, which is also a very recent tool, and are deploying these tools to investigate basic mechanisms of oncogenesis?
So yes, and as you know that AML is a very crowded field, and then the subsets of AML that we study, there are many people who are studying those. In fact, I saw that in your podcast there were people, like Yadira Soto, Feliciano, and others, and Yalido, who are doing absolutely fabulous stuff. And but the questions that we are asking are in some ways overlapping, right? So which is not a bad thing because AML remains to be a really bad disease, and 60 to 70% people are still not cured of AML, and so we all need to ask these questions. The thing is that when you're asking this question, you either need a completely different way of looking at the problem, which is a tool that is very unique and has not been applied before, or you need a different problem that people are not looking at to make your set sort of distinct, especially when you're starting out in the lab. So that's where we started asking this question. We started developing CRISPR libraries. Of course, everyone does that now, but when I started my lab, we didn't have any expertise in CRISPR, but I had spent enough time, making the conditional knockout mouse for AF10, which takes a couple of years to make the mouse and then a few months, if not years, to see if it has any phenotype, right? So the question was, if you could do this at scale and ask this question for every single epigenetic modifier, then you could just uh, find the ones that show the most interesting response, whether it's in terms of efficacy or selectivity. And so we said that let's build a library for all epigenetic modifiers. And then we ended up doing that, which led to some really cool results in my lab.
So when you say all epigenetic modifiers, what does this mean?
So, of course, you can never, the list of all epigenetic modifiers, we struggled a lot with that, because do we go with the epigenetic modifiers that are, all the usual suspects, the HDACs and the HATs and the KDMs and the CATs and stuff like that? Or you also add individual, like there are kinases that have been shown to phosphorylate chromatin. Do you add those? The the problem with doing that, like taking individual papers and adding everything that has ever had any connection to chromatin modifiers, then you end up with a list that is large, which is not a problem now. But then if that is one of your hits, then you want to see if it's a chromatin activity, which is like that one or two papers, those one or two papers, or it's traditional canonical kinase activity, right? So this was a challenge, and we did make a list of 600. I know people have now 700 or 800 proteins, and some even 1,000. But that's that's the challenge, right? Of making a definitive epigenetic library list. Uh, yeah, so that's how we ended up building a library. So one difference that we did is that we, based on Chris Vakoch's work, we tiled all the domains in epigenetic modulators, not just like 5 guide RNAs per exon 2, like some other people do. We tiled every domain with 5 guide RNAs. So if you have a protein that has 5 domains, let's say like KMT2A or something, then each of the domains has 5 guide RNAs. So we do see that it does show a better resolution of the data when you use a library like that.
Mm-hmm. You just said that you found interesting things. Can you tell us about those interesting things?
Yeah, so one of the interesting screens that we published in a paper 3 years ago now, 2 or 3 years ago, was we did a screen on. We wanted to know what are the epigenetic regulators of cell renewal. This is a question that has been asked many times by many people, but as you and other people in the field who know this and follow this, what used to happen in the last like 20 years ago or even 10 years ago is that you had some hints, right, that this protein like DOC1L or MLL might regulate Hox genes from Drosophila or from all the carefully done biochemical purification assays and other. And then you would make a knockout mouse, and then you would find that it controls HOX genes or other self-renewal associated pathways in leukemia. Then you would test if it does that in normal cells, and the usual very tedious but very cool, careful, year-long experiments that would be done. Now, what these CRISPR screens allow us to do is to ask this question: what are the epigenetic regulators of self renewal? Now, what do you call self renewal? And of course, there are many definitions, and so you had to use one single marker of self renewal, and that's an impossible thing to do in a phenotypic screen. So we just used this protein called MEIS1, which was tagged with GFP by Keith Humphreys. And so what we use this cell line, and the cell line is U937, which has a CAMF10 fusion that has almost defined all the work that I have done so far, starting from my PhD. My lab jokes, because we have many of our discoveries come from the cell line, and of course done in many cell lines and mouse models later. So my lab jokes that you didn't find U937, it found you. so, but so we used the U937 MEIS1 GFP cell line. We did the CRISPR screen, and we found a lot of epigenetic regulators that had been described by many different people in the field as regulators of Hox-MEIS genes, including MLL or KMT2A, ENL, or and of course DOT1L and AF10 and lots of others. So it just told us that the screen had worked really well, but it was kind of disappointing that there were hardly any new ones. And so there was, of course, SGF29, also called CCDC101. And I had no idea what this protein is at that time, and looked it up, and it turns out that it's a chromatin reader that Jerry Workman and Matthias Mann's lab and other have shown is a chromatin reader that reads K4 trimethylation and recruits the SAGA complex, the acetyl transferases GCN5 and PCAF, some of the oldest acetyl transferases known. I found it interesting that GCN5 was literally the first acetyl transferase discovered by David Alice in Tetra. So the SAGA complex is recruited to chromatin by SGF29, the chromatin reader, and so we decided to focus on that first. And we deleted the SGF29. We saw that it leads to a transcriptional downregulation of the usual suspects, some of the genes that have been causally associated with in leukemia, like the Hox genes and MEIS. But we also saw very strong effects on MYC, that MYC just goes down and it is lost from the chromatin fraction when you lose SGF29. And so we also did chromatin immunoprecipitation to see, chromatin enrichment proteomics, not chromatin. Of course, we do CHIP all the time, but chromatin enrichment proteomics we did, where that showed us that when you delete F29, there is a decreased abundance of CAT2A in the chromatin fraction and other members of the SAGA complex. So just also, again, showing something that Jerry Workman had shown, I think, in yeast. I could be wrong on which paper it was or whether it was Jerry Workman. I think it is. But the idea that SGF29 tethers the SAGA complex to chromatin. So that's what we found in the leukemia cells as well. Now the question was that we also got a lot of other hits like casein kinase 2, CK2. And again, CK2 was one of these where there was like one or two papers that have shown that Jeff Rosenfeld's lab had shown that it phosphorylates histones. And so that's why we included CK2. And so we found two components of the CK2 complex as being important for this. And so we have done a lot of studies on that as well, which show that. And the reason why we also focused on CK2 is because there is an inhibitor of CK2 that is in advanced phase clinical trials. And so that was interesting for us.
So imagine you are due to submitting a grant proposal tomorrow. What would you have written into this?
Well, again, I will repeat the same thing I said before, is that I will very likely go the safe route and use the preliminary data that we have from the Saga complex or from CK2 or other things that we have done recently, and write a grant on that, because people look for feasibility, understandably, right? So, can you do what you propose to do? So, we have a lot of that in the grant. And we will propose experiments that are a mix of the traditional, right? The A, B, and C. Here's what we will do, mechanism, etc., and innovation. And I just keep trying to push the boundary on the innovation. I sometimes have to restrain myself, because whenever we've come up with, we said something we want to do that is very unusual, we've unfortunately received criticism for that specific
Mm-hmm.
part of the specific aims. And so I have to be careful about that. But recently, our lab, because of our interest in gene fusions in general, has really become interested in other pediatric cancers that are driven by gene fusions. And so our latest story is on synovial sarcoma, which, as you know, is driven by this single gene fusion. The SS18 protein, which is a part of the BAF complex, fuses to SSX, one of the SSX genes. So my lab has been really interested in this, and we have some really interesting data on that. So we will be focusing on grants on that as well.
Okay, is this already published? Can you share something, or is it rather unpublished and you don't want to talk about it?
It is actually, we have received notice that it is accepted in principle. So I can share that it has been accepted in principle.
Do you
But
want to talk about it?
yeah, so I can tell you the story. We
Yeah.
always put stuff on bioarchive usually, because I think that that's just the right thing to do, to share it quickly with everyone. Usually it takes, as you know, months to years for the final paper to be accepted. And so that's kind of frustrating. So we have put that paper out in bioarchive. What we found is that, so our lab uses a lot, like others, of genetic screens, not just the ones we do, but the ones that have been done, like dependency maps, the depth map data. And so we saw that there were unique dependencies in synovial sarcoma that we could identify using depth map. And so one of these dependencies was SUMO2. And so we found through a series of in vitro, in vivo genetic and pharmacological experiments that blocking SUMO2 does interesting things. One is that it actually has strong anti-sarcoma effects in vitro and in vivo, but also it seems to regulate the fusion itself, which I'm very excited by. We've done, of course, Western blots, but we've also done cut and tag to see where the fusion binds, and we see a drop of fusion binding to its chromatin targets when we use SUMO2 inhibitors. We're still trying to figure out how that works. We don't think it's direct. We don't know that yet. We think it's indirectly affecting the non-canonical BAF complex that includes the fusion. But we are very excited by that result. And we have a lot of other ongoing projects in the lab that are very preliminary, where we are using these synovial sarcoma models to find more dependencies in this disease. Because, as you know, this is a terrible disease that has been very difficult to
Mm.
cure, and has a cure rate. I think 40% of the patients eventually succumb to the disease. And I am excited by this, by the research on this, because it is driven largely by this one single fusion, which is an epigenetic modifier. And these tumors do not seem to have any other mutations. So if you can find something that targets the fusion, then you can make an.
Yeah. Sounds very interesting. So for the last 45 minutes, we've been on a journey through your scientific career. And before switching gears, I want to ask, did we miss something important from the science side, or would you like to add something?
I'll just very quickly say that, you know, and also, of course, this is just because of the tremendous trainees that I've had, that we have done in the lab, uh, not just basic discovery of, like I told you, the different targets that we identified, whether in leukemia or sarcoma, and there are many others that I am not elaborating on today. But we discover them through CRISPR screens and others. But also, we go all the way in trying to make small molecule inhibitors. And so at the Sanford Burnham Prebys, they have this screening center, which is a very impressive operation where you can do small molecule screens on hundreds of thousands of compounds using robotics. And so what we have recently started doing is to build assays to find small molecule inhibitors for the targets that we identify. So in this lab, you get this unique opportunity. I tell my trainees, and this is not just an ad, this is not an advertisement. Uh, we get to work on, from finding the target all the way uh, to finding a small molecule. Of course, it takes a long time, this whole thing. So you may not be the person doing everything from start to end. But my first graduate student, for example, was involved in getting, you know, in identifying a target all the way to actually building an assay, a SEDSA assay to find small molecule inhibitors. And so this is a very unique experience, and I'm glad that we are at a stage now that we can do that for the targets that we identify. And of course, so uh, this is what really excites me the most, that you can go all the way from basic discovery to try and develop translational therapies. What really helps me a lot is that, as I mentioned to you, so Anaga, who is my wife, so she is the backbone of the lab. So she's a staff scientist, and she has been developing and some of the really important technologies, and some of the really cool stuff that is happening in my lab right now is being led by her. So that's really helpful.
And it's also a good team, right? You get to do all the things you like with the people you love.
Absolutely.
It's a good place to be in. So one thing I also wanted to talk about and ask you about is, because I just learned this a couple of weeks ago, that you are also a fellow podcaster and you started this. So maybe you can talk about the genesis and the motivation behind this project and how you find time in doing this.
I think the last question is the most difficult to answer. How do I find the time? It's really tough to find the time. But the reason I decided to do this podcast is because, first of all, as I had mentioned to you, I always liked reading and language. And I had this moment when I was asked by someone what I do, in the lab, and I described to them, and I realized that they didn't find it interesting at all, even though I thought it was really interesting. And I realized that the language, as I was mentioning, that we use is so dull and so boring, and so inaccessible to general audiences. I hate the word lay audiences, but you know what I mean, right? So it's very difficult to get across the messages. And then I realized, especially COVID, during that time, it was the turning point when I started doing these small videos explaining to people. Everyone was at home and they were wondering what is happening. And so I started making these small videos and, relaying scientific findings to people, right? So this is what the virus is, this is what it does, this is what you should do. And then I realized that people would still rely on social media, on Instagram and on YouTube shorts, Instagram reels for their, not for their entertainment, which is fine, but for their information. And I thought this is a big problem. And as we all know, this became a monumental problem, right? Because people just would not trust scientists. They don't think that, and then this seems to be growing, not just in the United States, but everywhere else as well, I think. And so I thought that the reason for that is that most of the scientists I admire, who I think know much more than I do, they are not even in the rooms where the conversations are happening. They are not on Instagram, they are not on YouTube or whatever, right? And of course, we almost, I am guilty of that as well. When a scientist starts simplifying science, uh, other scientists almost look down on, take that scientist as a non-serious person. I hope that changes because, and there are reasons for it, right? Because whenever you simplify something, let's say I say that, the DNA in all your cells is exactly the same. some scientists will stop you and say, actually, that's not true, because the brain cells have, and you're like, I know that, but when you try to convey a message, it has to be a simple message.
The majority
right?
is the same.
Right, exactly. So I think we worry about this all the time. Are we accurate? Are we? And I worry about that all the time when I do my podcast, right? Am I 100% right? And then I realize that people are taking their information from people who are not even 2% right. And all the scientists who I admire, I hope they listen to this, and because I want more and more of them actually to do what I am doing, because I'm sure they'll do a better job of this than I do. But many of them are not. Of course, they're probably just too busy. But I think more and more of us need to come to Instagram Reels and YouTube Shorts. I know we don't have the time. I don't have the time, but I've spent weeks and hours and days and nights trying to do that podcast, and I think it is very important.
Yeah, the thing is that there are different audiences, right? And I mean, you do your podcast, I do mine, and if you want to follow the scientific story and follow the PubMed record and things like that, then it gets very hard, and it's very, very time consuming to break it down to a level where you have the goal in mind and like the translationals and also the public audience. And for us, it's like more like the audience is people from the lab, probably because it's too complicated for people who are not from the lab. But what is the content that you are talking about?
So I actually wanted to talk about how scientific discoveries are made, because I saw that people just take things for granted, whether it's your CGM that you're wearing, even the eyeglasses that you're wearing, simple as that, to, the metformin that your uncle or dad just takes every day. You just take it for granted that it just magically appeared from somewhere, right? Nobody tells you the story of how that came about. And I think that it is important for two reasons. One is to understand the concept from the first principles. And the second is to appreciate, right? That, oh, okay, so this just didn't magically appear. It was people, dedicated people, spending their time in the very careful pursuit of finding the truth about life, about fundamental principles of life. And so I ended up, so the first episodes are on diabetes. And, it just happened that we started on diabetes with my colleague Pamela Itkin Ansari, who, if you've seen the podcast. And so we just decided to do an 8-part or 10-part series on diabetes. And we thought that it will be very interesting to actually just invite the people who have made the discoveries, and hope that they join us, because it'll be nice for people to hear from them. And so in episode 2, we have Herb Boyer, who founded Genentech, and telling the story of how they did it. We have Keiichi Takura, who did the first DNA synthesis ever of a human gene and cloned it, and he tells his story. So we traveled to City of Hope. We saw that. In episode 3, 4, 5, 6, we have interviewed incredible people. I don't want to give it away, but we have interviewed people who are really legends in the field. And so I'm excited about that. We want to tell the story of vaccines and of antibiotics, because I think they are very timely, as you know, very important. And I hope that people will engage and, come and join us, share the word, and share these stories. It's difficult, as you said, because these are 40-minute to an hour long. And so it's very difficult to get people to watch that. And one thing that we learned, Stefan, when we were doing this podcast, we always knew that if you're not a scientist or you're not a nerd, you want some entertainment, right? You want something that keeps you hooked to the show. So we did realize that. And then some of my favorite science stories involve human elements, right? When I read about Pasteur, it's not just about what he discovered, but, I was hearing Carl Zimmer's Airborne recently, and he starts with Pasteur's funeral and describes how diplomats from around the world came to his funeral. And so you're hooked, you want to see what's going on there, right?
Or how penicillin was discovered, right?
Sorry?
Or how penicillin was discovered.
Exactly. Yes. So one in the first episode, we told the story of Claude Bernard, this French scientist who is called the father of physiology, and his wife Marie Bernard was, worried about his animal experimentations, and she became this activist. And so when we told that story, I asked people, what was the thing that excited you? Because what excited me was that he found that glycogen is stored in the liver when you have too much food and used when you're fasting. And I thought, wow, that's interesting. He named glycogen. And what they found interesting is that his wife would not like his dog experiments. He worked on his pets,
Yeah.
and then they divorced because of their disagreements. And then I realized that you need to wrap education in entertainment. And so otherwise you lose the audience, right? So some scientists who are really serious scientists, they may think that this is kind of too corny, too much entertainment, but that's not our audience, right? We love if they watch it and share it, but we are trying to reach actually the general audience, because I think it's a very important, as important as I think it's the most important thing today.
But then this involves a lot of planning, right? Because the entertainment is really then the work.
Yes. Yeah. So this involves a lot of work. So we've been writing the scripts, and we've been getting people, interviewing people. And if we get a chance to actually meet them, so there is a person we interviewed. So Pam traveled to Oakland in San Francisco to actually meet, because some of these are 90-year-olds. They are legends in the field. And if you get a chance to meet them, you want to meet them, not just Zoom call.
That's true.
So it's a lot of work, but I hope, and that's why I'm hoping that it is shared by more people, not just just for reposting, but with thoughts. Because if they really like it, I think it should reach the maximum number of people.
Yeah. So
and we are not doing it for fame, because I'll tell you very honestly, I was reminded by people that I did not even introduce myself. We didn't introduce, I said, who are you? Why should I watch this? And then we started, putting in the names. So this is just simply purely for telling people the stories that we think are very important and exciting.
So where can we find it? Is it audio, video?
So it's on, we have gone to great lengths. As I mentioned to you, audio only would have been very much less work than video plus audio, because in the video version, which we have on YouTube, we have put in a lot of elements. When you talk about the Asilomar conference, we show the conference, we show Paul Berg's picture and Herb Boyer's picture and the discussions between Jim Watson and others. So we show those elements because I think that's really important. But I understand that not everybody, sits and watches a 40-minute long video on YouTube. And so what we've done is that the podcast is called The Discovery Dialogues. And we have it on YouTube with all the visual elements, but we also have it on YouTube as a podcast that you can listen to. We have it on Spotify, Amazon Music, and on Apple as well. So it's available. You can listen to it while driving in your car, which is what most people seem to do. And one challenge is that people don't get to the 40 minutes. I've seen that people watch 15, 20 minutes, maybe their drive is over or they get a YouTube ad or something. But I promise you that it gets better as it, as you go into the later minutes. And I hope more and more people do that.
And it's like 2 episodes right now?
It's 2 episodes right now, and we will have, I think, at least 6 more episodes on diabetes. We will tell the story of the continuous glucose monitors and sugar lows. We will tell and we'll hear it from the people who invented it. We will hear the story of cell replacement therapies, and hear it from the people who made major contributions there. We will hear, and I think what is fascinating in the next episode is we'll talk about venoms, which is one of my favorite topics of all time, and how it relates to diabetes. So we will end with what is the talk of the town right now, hopefully, is the GLP-1 agonists. And that's what we were going to end the diabetes episode with.
Perfect. So do we have a regular publishing schedule?
We do aim to put one per month. but it's been very challenging, with all the grad students and the, reviews and this and that, a lot of other, so I strictly do it just on my weekend schedule.
I see. Yeah, I will definitely have a look at it, and maybe also, I mean, it's audio and video, right? So you have both versions. I will check both of them out, and we'll happily share and put also all links into the show notes so that people can see and pass it along.
Thank you very much.
So thank you, Ani, for your time and for being on the show.
Thank you very much for having me.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.